Thank you so very much, particularly Oslim, for the kind invitation to this symposium, which is fantastic, and I have learned already a lot. I would like to step back. We have all heard already exome sequencing a bunch of times tonight, so I like to go a little bit through what it means and how it's conducted in a molecular diagnostic laboratory where I'm working. So this is my disclosure. In genetic testing, really, we have seen a revolution in the last few years. We have come from single gene variant analysis and sequencing just one single gene to going in sequencing whole panels of genes or even all genes in the human genomes that we can capture. So by tonight, I want to make sure that you understand what next-gen sequencing is and also newer technologies such as new deletion duplication testing by microarray methods and whole exome sequencing, and that you can differentiate what is the benefits and limitations of testing for large panels and exome sequencing, and what challenges they actually provide us with if we use them in a diagnostic setting and clinical practice. So at the center of all our attention really is that we have a patient and we like to help the patient and ask what is the cause of that genetic disease that we're suspecting, and of course how can we best treat it or prevent it. And in the past we have focused on single gene sequencing if it was a monogenic Mendelian inherited trait, or if it was a more multiple congenital anomalies, we went for karyotyping. However, not every time that we did those studies did we really very successful, and then what comes next? So usually we have scenarios where we have to look at disorders where multiple genes can cause the same phenotype, or the phenotype of a patient might be part of the spectrum of multiple different disorders. And in the end, we were like, okay, we really need to look at way more genes than what we have been doing before. And the tool in order to accomplish that really has been next-generation sequencing or massive parallel sequencing. It is an extremely high-throughput sequencing method, and it has been compared that one next-gen instrument probably can generate as much sequence as we used to do with our warehouse of capillary sequencing instruments when we started out doing sequencing. So this method allows you to sequence multiple genes at once. You can attack a whole entire pathway of different genes, or even all genes in the human genome. The sensitivity of that method has been very high. However, it is not as high, or at least in the beginning, we thought it was not as high as Sanger sequencing, which we consider our gold standard, which has at least 99.9% sensitivity. That is because we can identify most types of mutations with next-gen sequencing, but we had trouble with identifying insertions and deletions or duplications. So they were more problematic, particularly depending on the size of the product that we sequence, whether or not we sequence from both ends or just from one end, and so on. Also, we have problems with sequencing in areas where genes have pseudogenes, or there are regions of homology somewhere else in the genome, and the regions have a high content of Gs and Cs, which tend to be more difficult to handle. And not to forget, so sequencing instruments are quite expensive even now. To set it up is between $500,000 and even more at times if you consider all of the expenses that need to go into such an operation. And most particularly, it's bioinformatic resources, because this really makes next-gen sequencing possible. So to boil it down, next-gen sequencing, you start out with the patient DNA. You have only 2 copies of each gene, so we need to enrich the genetic material we want to look at and sequence. You can do that by PCR, like we have done in the past for regular ABI sequencing. So we can develop homebrew tests in our lab where we throw together multiple PCR reaction all in one tube, or it can be done more elegantly as by rain dense technology, which is a more microfluidic chip design. We can also use targeted enrichment using capture probes or baits that are DNA or RNA based and that are tiled across a template of the human sequences and are labeled, and with that label we can pull out those sequences that have hybridized to the complementary strand of DNA, and using one of those methods, we can enrich our target template. And then using a lot of biochemistry and chemistry for reactions, it actually, these reactions can be prepared for sequencing on a solid state, where basically you use the human DNA template strand, and then you synthesize a new strand complementary to that, and every time you incorporate a new nucleotide, it's fluorescently labeled, and you take a picture of the new nucleotide that's incorporated. You see that on the very far right-hand side, and then you generate millions of those pictures. They have to be overlaid and analyzed what's different between each of them, and that's where all the bioinformatics comes to play. So once you have generated sequence using next-gen sequencing, you get a lot of data and information, and that need bioinformatically analyzed, and I'm going to come to that in just a second. And then, of course, whenever you find something that you feel is important, you should, in a diagnostic laboratory, better confirm it and make sure that this is really what happens. So we use usually Sanger sequencing to confirm, but also to fill in any gaps of sequences that we couldn't reach with next-gen sequencing. And then, of course, whatever you found, you need to interpret, and that is not a small task. So bioinformatic analysis for next-gen data is pretty complex. So for every base that you want to analyze and look at and sequence, you get hundreds to thousands of overlapping reads that are aligned and staggered, as you can see here in the diagram. So if you look at red-colored bases, they are indicated by these colors that they differ from a reference sequence. So we compare all of those reads to the human genome reference sequence and indicate in our software where there are differences. So if every base is different across all of the reads, you would say this is a homozygous variant. If you find about 50% of the time a different nucleotides, then you would think it's heterozygous, and so on. So we need data processing and data calling of those variants, and we need to compare and annotate what those variants then mean by comparing to other databases. For example, looking into the SNP databases for looking for benign sequence variants in the general population, looking into mutation databases, what has previously been considered to be detrimental, and so on. And then we also look for novel variants that we have never seen before into different software algorithms where we try to predict, does it have an effect on the protein modeling or on protein structure or function? How is it conserved through evolution? And does it affect splicing and any other predictions we can make to classify whether or not a variant might be pathogenic and causing a disease, or more likely be benign, or some things that we simply cannot classify, we can't be sure. And those calls we call variants of unknown significance, or short VUS. So we can apply next-gen sequencing really by best using it for making panels of genes that we previously just did one by one because it was a heterogeneous disorder. Now we have an ability to look at whole panels of genes. We can design those panels really customized to include all of the genes that are very critical for a disorder, or we can include way more genes that we think there is some evidence for it that they could be in the same pathway, but there is no really clue and evidence that this is a disease-causing gene because nobody has found mutations yet. So typically, those panels tend to be more on the smaller side because we designed them to say we want to have panels that include genes that matter and that we actually can interpret the variants in. So typically, these are known disease genes where we have strong evidence for clinical utility, including those genes, which guarantees that we have the highest possible clinical and technical sensitivity for those panels. For example, most of the time, panels are designed to really cover every base of every gene, and if that's not possible by next-gen sequencing because you get a gap or it's an area you cannot easily obtain, then we can beef it up by including regular sequencing reaction and PCRing those areas so we have complete coverage. It is also desirable to perhaps combine the sequencing reaction with looking for deletions and duplications because, as we know, 1% of all our mutations we have are copy number variation and interrupt the gene. So some examples of next-gen panels are listed here, and there are really many, many more, and many different labs now offer those next-gen sequencing panels. Sometimes it's a kind of a number game fight. We have 5 more genes than you have, and that's why our panel is probably better than yours. It really comes down to what you use your panel for and whether or not you want to get a high number of variants of unknown significance and trade that in instead of having a small to be more inclusive. So the benefits of those targeted next-gen panels are that they usually have a high sensitivity, also that varies, of course, by what type of panels you have. It is definitely much faster and cheaper than sequential testing of single genes, one after another. You can make small panels or all-inclusive panels. It depends on your preference, what you like to use for a particular patient scenario. Depending on the design, you can design it so that you can work around pseudogene regions or homologous regions. Usually, if the genes are chosen very well and they are well-established genes, the rate of variance you cannot interpret is relatively low. And it's possible to combine it with concurrent deletion duplication testing to really get the most out of your panel and design. It has, of course, a lot of limitations too. For example, you have only the genes on the panels that you can look at, which means if there is a new gene coming out today in Nature Genetics, you cannot really put it right away into your panel. You need to redesign it and validate it, and so that takes some time. Also, deletion duplication testing cannot really be done at the same time with the same reaction. You have to add on another test and perform that separately, usually by microarray analysis. Also, you might incur some extra cost because you have to fill in gaps that you have from next-gen sequencing by ABI sequencing, and it might increase the turnaround time because you have to do extra work. And also, it is a very phenotype-driven targeted design. So if a patient has a more complex phenotype that doesn't fit really a narrow disease, then the benefit of such a panel is limited. So I want to very briefly touch on deletion duplication testing. So that is a method that has emerged coming from chromosomal microarray analysis using oligonucleotide arrays. There are different other methods that can be used to detect deletions in part of a gene or an entire gene, such as real-time PCR or MLPA. But exon-level array is just more efficient for most of the genes that we can analyze. In the diagram, you see the 2 copies of a gene, and in one of the copies, exon 2 is deleted. So for array-CGH, you really compare the hybridization signals of probes that are on both copies of the gene in your patient to that signals that you get in a normal control. And if the patient has a deletion, as shown here with the green probes, that means you get in your reaction a higher signal from the controls because it's missing half of the signal density from your patient, and that's how it can be detected. This method is very elegant, very reliable. It allows you to detect very large deletions as well as tiny deletions. So in our lab, we use a cutoff of about 250 kilobases as a lower cutoff for calling reliably small deletions within a gene, usually 1 exon. And you see on the left-hand side a deletion in the BRCA1 gene of just 1 single exon. And on the opposite side, you see a much larger deletion, obviously much easier to identify. So this technology allows you to reveal mutations you definitely would miss by regular sequencing. It's very important for dominant disorders that are caused by haploinsufficiency, as many of the cancer genes, and I listed you some examples here. But it's also important for X-linked disorders. For example, in ectodermal dysplasia, we found that 10% of patients have deletions. Or even in recessive disorders, where you sometimes really find only one disease-causing mutation by sequencing, and you were left with uncertainty, is that it or not? And if you address exon array deletion testing, you might find the second missing mutation, which helps you to do carrier testing in the family or offer prenatal testing. So as an example of how you can use this panel testing for this audience, for this program, I just want to quickly address our results for using a SCID panel we have offered. It contains 18 different genes, and we broke it down for ease of use based on other previous diagnostic tests into a B-cell negative SCID panel and a B-cell positive SCID panel. All in all, we tested 87 patients with that panel. It included next-gen sequencing as well as del-dup testing for one of the genes that is known to have more often deletions identified. So this is the outcome for those subset of patients we tested with this panel. And as you can see here, you not only identify positive and negative cases and not as many positives as you would wish, but at least we have close to 40% for the B-cell negative SCID and about a third of the patients with the B-cell positive who have a mutation. I want to draw your attention to the big blob in the middle. These are the patients who have variants of unknown significance. These are the ones that we just cannot identify with certainty whether or not they cause a disease or not. And this is one of the reasons why testing panels, and you have to be sure you understand and can accept that you might get a result that is not just black and white, that is gray. This slide shows you the overall sensitivity for each one of those panels, and as you can see, it varies by indication. The comprehensive SCID panel shows a lower percentage of positive rate, but that, I think, is due to the very inhomogeneous diagnosis and groups of patients who get referred for this panel. So if you cannot really drill it down any more or any narrower in B-cell positive or negative cases, you have a very heterogeneous cohort, and this is less likely to show positive results. So we do the next-gen sequencing panels, we do deletions, duplications testing. It still leaves you with a good number of patients where you simply don't find your answer. And whole exome sequencing has emerged as one of the new diagnostic tools to help you identify mutations in genes you couldn't identify before. So to boil it down and make it really simple, exome sequencing is sequencing of the coding genome, or those parts of the genomes that encode proteins called exons. And we know this is important because the majority of those mutations that we have so far identified to cause a disease are localized in those areas. So as we heard before, it represents about only 1% of the whole entire genome. It comprises 20,500 genes or so and about 180,000 exons. It includes also coding sequence, the flanking splice sites, but it does not include untranslated regions and regions of genes that show duplications or homologies. So it has some limitations. It allows, whole exome sequencing allows you to simultaneously sequence all of the genes in the exome you can capture. And we use capture probes for that, as I explained before, usually DNA or RNA probes. It generates, obviously, a massive amount of data, and it needs a lot of filtering and looking down to find really those changes that could be causing the disease in your patient. It is much less expensive than sequencing the entire whole genome, and for that reason, I think it was called the sweet spot before we can do whole genome sequencing. So for which patients should you consider that? So we started out using exome sequencing for patients who have undergone an extensive diagnostic odyssey where we did a zillion of other diagnostic tests and simply over years could not even find what's causing the patient's condition. It also can be used for patients with the disorders that can be explained by many, many different disorders, such as intellectual disability, developmental delay, hypertonia, congenital heart defects, or also congenital anomalies. Often we also get referral for patients who have a very atypical disease presentation, or for patients where they have very likely a very, very rare specific disorder, but there is simply no testing available right now. So if you compare that to the gene panels really very quickly, obviously exome sequencing gives you way more material. You have all genes you can look into. We are using capture, and there are more methods, different methods available for panels. We use next-gen sequencing for both applications. For whole exome sequencing, you have to accept that there are gaps in the sequence. You are unable to get every single nucleotide of every single gene. That's impossible at present. So we have to be comfortable with some areas simply not being evaluated. Obviously, also, it has a much higher cost, and it takes a much longer time to get to results. So this diagram shows you how we are conducting exome sequencing in a diagnostic laboratory, just the workflow of how a sample flows through the lab. You start out with a sample coming in from the families. We go through all of that. What I explained to you goes into next-gen sequencing to prepare the libraries and sequence them. And then we have that enormous bioinformatic filtering process that needs to take place before we can look by eye and review the clinical data and compare that with what we find in order to identify any smoking gun or variants that we believe have something to do with the patient's phenotype. And those will then be confirmed, and we do segregation studies in the family, see if that makes all sense, and all these data get reviewed again, and then we report the findings for everything we can identify related to the patient's phenotype. In addition to that, obviously, if you have the whole entire genome to look at and you see every gene, there will be variants you find incidentally that have absolutely nothing to do with your patient's phenotype. For example, mutations that are previously being reported with hyperthermia or cancer predisposition. And those have to be evaluated, and we have to come to terms of what we do with that, whether or not we report it to the family or not. So we need to discuss those findings and decide. So the challenges are multifold for exome sequencing, starting with data filtering and interpretation. So on average, you have more than 150,000 variants that will be called and identified, and we have to filter it down to the 1 or 2 or 3 that are important for the patient. So we look for the frequency of those variants in the general population and filter out polymorphisms. We look for databases with known mutations and filter out things that are already known and determine if that disease has anything to do with the patient's phenotype. We also look for genes and pathways that we know might be related to the patient's disorder. We look for detrimental sequence changes like loss of function mutations first and filter those out so we can look for the worst scenario first. And of course, if we can, we use other approaches to narrow it down so we don't need to look at the whole entire genome. For example, if you have consanguineous families, often it helps to look at blocks of homozygosity. And also, in addition, we can look for areas where we have a lower number of reads and coverage than we normally would have, and that might give us a clue to maybe seeing deletions, duplications. That algorithm is still in progress, and it's very unreliable, but sometimes you can make a call. And when we then try to identify the variants that matter, really we are posed by a lot of challenges and interpretations because we know so many disorders have reduced penetrance or variable expressivity, and you can find a variant in an unaffected parent and you cannot exclude that variant based on that fact. Also, there are epigenetic factors which we cannot include in this algorithm, or phenocopies, uniparental disomy, or the fact that females might be affected if there is skewed X inactivation for X-linked traits. So once we looked through all those variants and filtered out what we believe is important, the different types of mutations that we identify are listed here. So usually if we are very lucky and it's a good day, then we find immediately something that is known and has previously been published or looks and is expected to be a pathogenic mutation causing a disease that totally explains the patient's phenotype. So if that is the case, then we definitely do a happy dance around in our laboratory, and you hear people yelling and say, yeah, I found it, happy dance, and that's how we do it. However, most of the time, we come up with variants yet you cannot conclusively classify will cause a disease, or the variant is in a gene where you are not sure that this is really going along with the entire phenotype for the patient. And Sean's examples that she showed us this, a while ago this afternoon was one of those examples where there are multiple variants in two genes that we simply could not classify whether or not they have anything to do with the patient's phenotype, but they might explain some of the features. So if you find those variants of unknown significance, what are you left to do with? We can only recommend to follow up on that to try to see if there is any research information available, if there are animals or pathway studies. It helps to look for patients who have similar features or variants in the same gene, and to connect those across the country, across internationally different countries, and maybe reanalyze and rerun the data that we have now in the future, because these are not static analysis every day. There's new information coming out, and information might change over time. And then in very rare instances, we come across what we call a smoking gun, meaning you find a detrimental variant or change, like a nonsense mutation or frameshift mutation in a gene we have never seen or heard. There's no information available in any of the databases that gives you even a hint or a clue in what pathways this could be fitting in, or what clinical features it might be associated with. Many of those changes have been shown, particularly on the X chromosome, to be totally benign and found in a high percentage in normal controls. But for genes that you simply don't know anything about, you can make no assumptions. So then it helps if you can collaborate with other labs, and there are collaborations going on internationally on a large scale, so that we can try to pull together different patients who have all variants in the same gene, and then after you've seen it 2 or 3 or 4 times and they all seem to present with similar features, then it's able to you are able to make a new diagnosis and find a new disease gene, as was shown here previous for the immunodeficiencies, for example. So if we report our findings, usually the reports go out as a positive report if we can conclusively determine this is causing all the features of the patient as reported to us. This is something you can take home and go to the bank. We offer prenatal testing on those variants, so these are conclusive diagnoses. A part of the patients have negative results, which seems very odd or strange if you consider that we look at 20,500 genes. But if we cannot identify anything that really can conclusively or even vaguely be related to the patient's phenotypes, then we elect not to report it, because it will not help your patient in the long run. We need to keep an open mind. And the majority of patients, as you see in the diagram, really about half of them have variants where we feel like this could be something, but we are not sure, or we find one mutation in a recessive gene. We don't know, is that a carrier, or is that really the diagnosis and we were just not able to identify it, or is it one of those new smoking gun genes that later will come out to be really disease-causing? We just need to get more information. So very briefly, we have heard examples of successful exome sequencing before, so I can make it very quick. This was a case with a severe combined immune deficiency. with predominant T-cell defects, but elevated natural killer cells. There was a history of recurrent skin infection, otitis, pneumonia. The patient also had a stroke, and it was the rest of the family was unaffected, and we received only the patient sample for analysis. We didn't receive both parents. So when we looked at the sequence results that we obtained, we filtered out all the variants that were detrimental. We looked at things that are homozygous or de novo, and we didn't identify any good candidate gene. However, when we looked at multiple hits in one and the same gene, there were 2 variants found in RAC1, which is a known immunodeficiency gene causing Omenn disease, and SCID. So we found 1 mutation previously published, which gives you a good idea that this is a good hit. And the second variants that we identified, these are the ones that are highlighted in yellow, was a novel variant. However, there were mutations all around it. All prediction models showed in red here that predicted a damaging effect on protein function. So obviously this is a very good candidate for being the solution. However, if you find 2 variants in a recessive gene, you still have to check parents and make sure they are not inherited on the same allele, but they are present in trans on different alleles. So that is what needs to be done in order to conclusively make the diagnosis. And very briefly, another case of Sjogren-Larsen syndrome shown here is this is a disorder that presents with congenital erythroderma, meaning redness of the skin and scaling. But later on, the child did not do so well. There was developmental delays and constipation and hip dysplasia. And on MRI was shown periventricular white matter changes, which is pretty serious. So after some other tests, exome sequencing was done, and revealed 1 heterozygous frameshift mutation on the paternal allele, which was confirmed, but that left still the second allele to be determined. So we looked at the sequence data and coverage, how many reads we have, and we discovered that for 2 exons, exon 2 and 3, that are highlighted in red, we really didn't have as many reads as we normally have compared to all the other exomes that we have from other individuals. So we suspected there could be a small deletion in that gene. And lo and behold, it was confirmed by using exon level array analysis. And as you can see, it extends into exon 1 and maybe beyond. So this was on the maternal allele, a small deletions that is a second detrimental mutation in this patient. And the patient has Sjogren-Larsen disease with progressive white matter disease and due to fatty aldehyde dehydrogenase deficiency. So all of this was what we have done when we do exome analysis, looking really targeted to the patients or screening for prophylaxis or treatment for very serious disorders, and could have really serious consequences for an asymptomatic person it's identified in. So childhood or adult onset cancer syndromes, cardiac disorders, neuropsychiatric disorders, it's a long list. Also, we see information about the carrier status for recessive disorders or for pharmacogenetic variants, such as for warfarin metabolism, or susceptibility alleles for complex traits. So the question really is, how much and what of that information that might be revealed really is being reported to the patient, and how much does the patient want to have that information, or the physician who orders that test? So the American College of Medical Genetics has put a lot of work and effort into making recommendations how to deal with that situation of incidental findings, and came up in March of last year with some recommendations. For example, the lab must report constitutional mutations found in a minimum list of 56 different genes, regardless of the indication for sequencing or the age of the patient, meaning disclosing that even for children. And as of the March of this year, now we allow also for an opt-out of that, so patients can say, no, I do not want to have those results disclosed. These variants are those that have previously been published and meet a very high threshold of being pathogenic. So we usually do not include anything where we have even the slightest doubt that this really could be pathogenic. And obviously, providers must provide comprehensive pre and post testing counseling to the patients in order to discuss those findings. So here is a minimal list of the 56 different genes. You see the disorders they are associated with, a large list of cancer syndromes, of cardiac disorders that might go along even with sudden cardiac deaths. So there are serious conditions in here. There was a big debate whether or not this is the best list or it can be improved, and the college definitely has made it a mission to review those genes regularly and to take suggestions of how to improve it. So they suggested that those variants would probably be identified in about 1% of all the patients tested. Well, over time, we have learned that this percentage is quite higher, so it ranges between 6 and 9% for the different labs who reported findings. So you have to be comfortable with receiving those results when you do diagnostic exome sequencing. There are also some differences between the labs that provide exome sequencing. So for example, you can just test the proband alone, as in the one example I showed you first with SCID. And this is usually cheaper because you only sequence one person. However, it's not possible to immediately determine if you have a de novo mutation or a compound heterozygous mutation. Trio analysis tends to be the most efficient way of using your money for exome sequencing because those types of mutations can be identified and de novo changes comprise a large percentage of the positive findings. It also makes analysis faster and you have fewer variants that you cannot interpret because you can use segregation analysis in your family. For example, based on the strategies, whether or not you include proband or not, or parents or not, you see here's the differences in the yield of exome sequencing. So if you do trio analysis, it tends to be between 31 and 32% positive findings. 25% was reported by Baylor labs who do proband analysis and then later on do segregation studies by like carrier testing by ABI. And 19% were positive findings for looking only at de novo changes in patients with epileptic encephalopathy. That is a study performed by the EPI4K consortium. Other differences between labs might be that some labs offer exome sequencing alone, others combine it with mitochondrial genome sequencing, which might be a good supplemental approach for those patients who have a mitochondrial phenotype. And then, of course, there are labs who report only the phenotype-specific findings, and others might report carrier status or pharmacogenomic risk alleles. However, and I really want to make that point, even if time is running out, it is extremely important for any exome analysis that you provide us with clinical information and family history so we can use that information to the advantage for filtering out variants that matter and that are important. So with that sentiment, I like to conclude and thank you all for your attention and hope we can discuss it more tomorrow or tonight. Thank you.